This week I feel like we're coming full circle. I've been going back through all the content I posted for you over the past 12 weeks to plan for your final week of assessments and to make a list of all the documents and charts we made that might help you do well that last week. We started our journey together talking about cells and what they are and how viruses fit into the whole scheme of life. Yes, there are many compelling reasons to exclude them and keep them out of this framework of living things. And yet here we are, almost at the end of our course, talking about viruses again. I went through the Look What You Did section, and one of the first things I posted there were some of your opinions on how to handle viruses. Do you remember that? Anyway, I think some of you are absolutely right. We're going to be forced to broaden our idea of what life is because we're finding more and more evidence that living cells and viruses are deeply entwined and have been from the very beginning. This week we're devoting to new therapies, and each one of them involves viruses. We're going to learn about CRISPR, a genetic editing tool that was discovered in bacteria, and its purpose seems to be to protect the bacterial cell against invasion by random viral DNA. The process is so efficient, it's downright scary. We can literally cut, insert, and correct any section of DNA we want in any type of living thing. Let's think about all the ways that could be used, both good and bad. You will see a movie from the Howard Hughes Medical Center about a little girl whose vision was restored using gene therapy. Gene therapy is only possible because of viruses and how good they are at getting into target cells. In this case, the blindness was a very rare type of genetic mutation that both parents had. The virus delivered a functioning gene that corrected for this vision defect. The last therapy we will focus on are bacteriophages, which are viruses that infect bacteria. The thing about bacteriophages is that they're extremely specific. In order to use them to treat human infection, you need to know exactly what bacteria is causing the infection, and then give the phage that can enter and destroy that one bacterial species. Using bacteriophages does not eliminate the need to culture out the bacteria to identify the cause of the infection, but it's like using a sniper to kill a bad guy instead of throwing a bomb into a building and killing everyone. We're going to consider antibiotics to be the equivalent of bombs someday because they're so nonspecific and they eliminate so many good bacteria along with the bad. You have your very last Teach the Class quiz this week on chronic and brain pathogens. Same pattern as always. Review the presentation, print out the slides as a reference, and when you feel confident, take the quiz. Our notable scientists for this week are so timely. The two women who won the Nobel Prize in 2020 for discovering CRISPR. This award is so important because it was the first time two women shared the Nobel Prize ever. It was also significant because we've learned about scientists who fought with one another or tried to claim all the glory for themselves. But we have also seen collaboration where the discovery might never have been made without multiple people freely sharing their work and what they learned. I think CRISPR is a perfect example of a discovery that might have taken much longer without the input not only of the two Nobel winners, Jennifer Doudna and Emmanuel Carpentier, but their graduate students and staff. The videos I posted are a little longer than I would like, but there's something magical about hearing a scientist explain her research in her own voice. I know it's Thanksgiving week at the end of a terrible, heartbreaking year, but I hope you can find something to be thankful for wherever you find yourself on Thursday. our table will definitely be a little more empty than it usually is. If you do find yourself with some free time this week, I think the best thing you can do to get ready for our final week is to go back through all your charts, both the ones you made and the ones I made for you. There's some great material in the Look What You Did section, and all your Teach the Class handouts will be helpful. This course was never about memorization, so try to organize this material into a system that will work for you. Don't try to memorize anything. I will do one more round of Collaborate sessions during week 14. That's going to be the week that we get back from our Thanksgiving break. Not that we get a break because we're online, right? We don't get breaks. Anyway, I'm going to give you the opportunity to come to another Collaborate session and by then I'll have some more definite material posted for you so you can see exactly what you're going to need to do. And there won't be any new material posted that final week. It's just going to be assessments. With that, I'll sign off. We're so close to being finished, and I'm so proud of you all. I know there were times when you wanted to quit and wondered if you could get through it this semester, but here you are. I am thankful for you. I hope you have a happy, restful day with the people you love the most, even if that's through a computer screen.